Okay, well, welcome everyone to our first Coeliac Disease webinar series hosted by Wesley Research Institute. My name is James Daveson. I'm a gastroenterologist. Before we start formally, I just wanted to acknowledge the Jagera and Turrbal peoples, the traditional custodians of this land on which we meet and pay our respects to their elders past and present, and to any attending today. Like I said, my name is James Daveson. I'm a gastroenterologist. I work clinically both in Brisbane and also up north in Mackay, which is where I grew up. When we thought about doing this webinar series, the main reason why I thought it would be a good idea is we've been involved in clinical trials for celiac disease now for the last 15 to 20 years. And back then, there weren't a lot of clinical trials for celiac disease. There weren't a lot of therapies around. So we got to know the people behind the studies really well. And so did the trial participants, and so did the people in the celiac community. And it felt like we were all on the same journey together. Now, there are multiple clinical studies for celiac disease. And I would like people to try and have that same opportunity. So the idea was that we would invite people from the biotechs and behind these new therapies for celiac disease and get to speak to us on a webinar. And then we thought it would probably be a good idea to have a clinical part just before we invite them to speak as well. This is the first of hopefully several, and we'll be inviting expert clinicians and researchers to try and give us their insights on the new updates in celiac disease. Anyway, we've had over 1,200 people register for this webinar, so clearly there's an interest, and hopefully it will be of success. So Dr. Jason Chien is the Chief Medical Officer at Mozart Therapeutics, and he'll be joining us just after I finish my talk, and I welcome him today. I'll tell you more about him as we get to his presentation. Wesley Research Institute is the second oldest research organisation in Queensland. It's located at Wesley Hospital. It conducts research across multiple themes. It's not for profit and it relies on people to support it, which we do through the generosity of others. So if you want to find out more about Wesley Research, there's its email address. I'd also like to very much thank the Rosalie Martin Memorial Fund, without whom this would not have been possible. So the fund was set up in the memory of Rosalie Martin, AM, who was a board member at WRI, thanks to the generosity of her family, who really would like the fund to see information disseminated to the community, like we're trying to do tonight, and have supported many events that we've had at WRI. We'd also like to thank Coeliac Australia, our partner, for collaborating and supporting this series, most of whom who have celiac disease already know about and value. So, moving along, my talk's about the pathways to treatment, and I really just wanted to set the scene in this talk so that we can really get a lot of value from people like Jason, new treatments. So I guess the first question is, why do we need a treatment for celiac disease? How do we find one? Where are we up to with that? And how do we develop a therapy? So why do we need a treatment when we already have a treatment in the gluten-free diet? And I think the first thing to note is this is quite a common disease. So if it affects 1 to 1.5% of Australians, and really we're talking nearly 400,000 people in the country with this disease. And in Queensland, that's around 80,000, which obviously increases to nearly 120,000 in New South Wales, our most populous state. And that's just in Australia, but globally, equally, we think it affects about 1% of the global population. So in the United States, that's somewhere between 2.5 to 3.5 million. And if we ask people who have celiac disease if they're interested in finding a medical therapy, then the great majority are. 80% have either a moderate or a great deal of interest in finding a therapy outside of the gluten-free diet. And if we ask people with celiac disease how satisfied they are with the gluten-free diet, the minority feel that they are satisfied with a gluten-free diet for the disease. And celiac disease causes symptoms. So most people probably still think the main symptoms of celiac disease are diarrhea or abdominal bloating or failure to thrive. And that certainly was the case. So this is a child back in the 1920s, before we knew that gluten caused celiac disease, and they would present and be malnourished and waste away. And there was a mortality rate associated with this disease. As we've gained access to better ways of diagnosing celiac disease, we understand now that it presents in a large number of other ways. So whether that be with bone fractures from osteoporosis or unexplained fertility or problems putting enamel on our teeth, they are all ways that people with celiac disease present. Iron deficiency anemia is a very common presentation of celiac disease. Dermatitis atopiformis is an unusual skin manifestation. Not only does it cause symptoms, at times celiac disease becomes refractory to removing gluten from the diet. And then those people can go on to develop intestinal lymphoma. And treating celiac disease improves the outcomes. The other interesting thing about celiac disease is the symptoms can change. So people may present with symptoms of celiac disease being lethargy or brain fog or not feeling quite right. And if we put them on a gluten-free diet, those symptoms do tend to settle down. But after 1 to 2 years, they start to develop a different set of symptoms, of which vomiting is one of the common ones. And I think that vomiting has led to this perception that the difficulties they have with eating out, and it can lead to social isolation. And that person in the bubble probably has a good reason to be in that bubble. And they're the sort of people that go to a restaurant and ask, is there gluten in this? How have you prepared it? Has it been contaminated? Because if they do eat gluten in their diet, they can get quite sick. And recently we've come to understand why that occurs, or we've at least validated their symptoms. So if you have people with celiac disease on a strict gluten-free diet, which they are after they are diagnosed, a percentage of those people after a couple of years, if they are exposed to gluten, within around 1 to 2 hours start to vomit. And with that, we start to see their interleukin-2 levels go up. So interleukin-2 is an inflammatory cytokine. And it's important to mention it because not only does it cause symptoms, which we look to try and control in clinical trials for celiac disease, but we can also use it as a biomarker to see how well some of these treatments are actually working for people with celiac disease. And we also know that the intensity of that cytokine release can correlate quite nicely with the severity of their symptoms. The treatment burden of a gluten-free diet is also very high. This questionnaire was sent out to a number of people with different diseases. So here we have someone with high blood pressure. Here we have someone with end-stage renal failure. And if you ask them, what's the burden of this disease in terms of how easy it is to control? Obviously, people with renal failure who have to come in for dialysis 3 times a week find that burden very high. People who have to take a tablet for reflux or hypertension once a day may not find that burden too high. But interestingly, celiac disease came in second. And that's probably because people with celiac disease have to continuously think about what they eat, day in, day out with every meal. A gluten-free diet is also high in fats and sugar. If you take out the gluten, you have to find something to make it more palatable. It's expensive. There's no Medicare rebate for this treatment. It's very difficult to maintain a gluten-free diet when you're traveling. It's very difficult for adolescents who are trying to meet and expand their social horizons to follow a gluten-free diet. For the financially vulnerable or cognitively challenged, or even living outside of a metropolitan area, a gluten-free diet can be very difficult. And almost to add insult to injury, we also now are beginning to think it doesn't work as well as what we may have thought it did. So this is Australia, New Zealand, and the United States. And these were their biopsy results when they entered the trial. So above the red line, all the biopsies are normal, below the red line, all the biopsies were abnormal. So if you look at the total columns for Australia, you'll see that only 4 out of 39 people had a normal small bowel at the time of entry into the study. Similarly for New Zealand, only 1 out of 8, and in the United States, 6 out of 78. So 91% of the people entering this study, who we thought had been on a strict gluten-free diet for 2 years with normal serology, had actually failed to normalize their small bowel despite being on a strict gluten-free diet. So we need a treatment outside of a gluten-free diet. So how do we go about finding one? And this is probably the most important part of this presentation, to try and set up an understanding of where we're working out where these treatments should be targeted. So whenever we go to a talk with these cartoons put up, and when I see them, by the time I almost get my head around it, they've moved on and I'm lost. And so what I really need, what I'm going to try to do today is go through that slide in a cartoon type fashion, so we can all move forwards together. So a protein is made up of amino acids. Amino acids joined together form peptides, which are short chains. And in proteins, we have lots of different peptides, and proteins are very long collections of amino acids. And gluten is a protein. It's a mixture of hundreds of proteins. And what we do when we eat gluten is we have to break it down and get it back to these individual amino acids so we can absorb it. But humans struggle to do that. Whether you have celiac disease or not, humans can only break gluten down in parts to these peptides. And it's these peptides that people with celiac disease see as very dangerous and toxic to them. People without celiac disease see that peptide and aren't too bothered by it, but people with celiac disease see it differently. So now I've drawn a picture of that gluten protein, and some people in the audience will already have seen that there's some collections where there's green and blue amino acids. So when we find these together, which are proline and glutamine, the bonds between them are very strong. And those are the ones that humans have trouble breaking down. So when they ingest it, those peptides are left intact, whereas all the other amino acid bonds are broken down and they get absorbed. So we talk about these peptides being toxic to people with celiac disease. And when they ingest them, they develop an immune response to them. So they're immunogenic to people with celiac disease. So they're what we call toxic immunogenic peptides. Interestingly, when we eat these peptides, if we don't break them down, we can find them in our urine and stools. And that's been the focus of one of the studies that I'll talk about a bit later. So now we go back to the bowel wall. So this is the gut lumen here, and this is the lamina propria. So inside the body and outside the body above it. And they're all those little villi that we talk about with celiac disease. Now we've got the mouth in front, and what we're doing is we're eating our gluten. And then in the gut lumen, it gets broken down to those toxic immunogenic peptides. And I'm not going to keep drawing those because that was just too difficult. So I've represented each of those with a little blue dot. So we'll get rid of those now. So each little blue dot is one of those peptides. Celiac disease, one of the things that happens is a bit leaky, more permeable. So these peptides go through the lining of the bowel, either between the cells or through the cells. And we start to get the release of inflammatory cytokines like interleukin-15 that draw other cells into this lining of the bowel and start to break it down and hurt it. But these little gluten peptides, they chug along in the lamina propria, and one of the first things they encounter is this thing called transglutaminase 2, which I'll probably refer at times to Pac-Man. So the transglutaminase 2 gets hold of the gluten peptides, and what they do is they deamidate it. Well, they give it this really strong negative charge and they change it a little bit. And that's really important because then it gets presented to other cells in our immune system. So these gluten peptides then go down, and this cell called an antigen presenting cell sees it. Anything that's foreign to the body that comes into the body that results in an immune response being generated is called an antigen. So these deaminated gluten peptides in people with celiac disease are antigens, and they're being presented on these cells. And one of the interesting things clinically is that little molecule that presents that deaminated gluten peptide is coded for by our genes. So if we don't have the gene, we don't have the molecule, we can't present the peptide. So it's very unlikely we'll get celiac disease. This is someone's actual HLA result for that molecule. And as you can see here, it's not detected, so they can't get celiac disease. Then the star of the show, these CD4 pathogenic T-cells see that deaminated gluten and they bind to it. And then those CD4 cells proliferate, which I'm sure Jason will talk to you about a bit later. B cells are stimulated, they also proliferate. The T cells release more cytokines. That's the interleukin-2 that we spoke about before that cause symptoms. And that's important for clinical trials because if we can turn off that pathway, we can reduce the amount of interleukin-2, we can reduce the symptoms. So we will talk about interleukin-2 a bit later on as well. And then these B cells make antibodies, and the antibodies go looking for the deaminated gluten peptides and the transglutaminase. So it's very much, celiac disease is very much an immunological reaction. We still think, a lot of people still think of it as a gut disease, but as you can see, there's this florid immune response happening. Those antibodies are the things we use to diagnose celiac disease. So again, this is someone's result. So less than 7 means they don't have any of these antibodies floating around. Or if they're in remission, they've completely avoided gluten now, so they don't make antibodies because there's no gluten peptides coming through. So unfortunately, I can't see everyone's faces to see if they're following this or not, if I'm doing a good job, but I think it's really important that we keep that in mind as we talk about some of the therapies. So if we know that's how celiac disease occurs, we can direct our therapy to different areas of that pathway and see if we can turn it off at certain points. So we can talk about the gluten coming into the gut lumen. So if you remember those little peptides that are chains of amino acids, if we can break them up further, they'll get absorbed and no longer be toxic. So that's one approach. Or we could genetically modify the gluten so it's no longer toxic to people with celiac disease. Or we could bind that gluten and have it pass through the bowel so it doesn't get absorbed as much. So reducing the gluten load. We could try and repair the damage that's occurred to that bowel wall. And we'll talk about that study a little bit later as it's been recently published. We could have drugs that try and block the Pac-Man or the transglutaminase so that they can no longer deaminate that gluten, so it's no longer as toxic to people with celiac disease. So we could block the immune response that way. We could try and turn off some of these cytokines, these inflammatory mediators of damage to the bowel wall, which people are trying to do. Or we could block this little molecule that binds the gluten. We could also look at ways to deactivate these pathogenic T cells. So they're populations of T cells that sit in people with celiac disease, and they proliferate and are activated when they're exposed to it. But they're not supposed to be doing that. It's an aberrant immune response. Or we could try and teach these T cells that gluten is actually quite harmless, and we could try and re-educate them so that they no longer see gluten as being dangerous to people with celiac disease. So there's many drugs now that are looking at various parts of the pathway to try and turn off this immune response in celiac disease. So where are we up to? We have all these potential candidates, but where are we actually up to? This is a little timeline I put together. So 10,000 years ago, humans developed the ability to harvest wheat. And that allowed them to form communities and set down roots instead of being hunters and gatherers. Just as an indication of where we're up to, I thought I'd throw in the pyramids from 6,000 years ago. And then 2,000 years ago, the first description of celiac disease was made by Areteus. It wasn't for another 3,500 years until Samuel G in London, who was able to read ancient Greek, wrote the first modern description of celiac disease. And he took the word that Areteus is used in Coeliacos and translated it to English as celiac. And that's how we came up with the name of celiac disease. But it was around 40 to 50 people knew it was something in the diet that was making them sick. So the first treatment started to evolve. They didn't know what it was making these children sick, but so they put children on bananas only, and they found they got better, but they still didn't know it was gluten. It wasn't for another 20 to 30 years that Wilhelm Dick in the Netherlands identified wheat and gluten as the thing in the diet that was making these children sick with celiac disease. And so the gluten-free diet evolved, and really that's been the only treatment we've had since, nearly 80 years later. Interest only, people started to think about treatments for celiac disease. We had our hookworm trial nearly 15 years ago now. And then ALV-003 around the same time was that one that looked at trying to break down those peptides so they were no longer immunogenic, and has now been the most advanced therapy for celiac disease. We also had Imisan T come forward with the celiac vaccine, and most people in Australia will be familiar with this because they were very involved in the clinical trial for the celiac vaccine. Unfortunately, neither the worms or the vaccine are probably going to be a therapy for celiac disease, but we did learn a lot from them. The whole story about interleukin-2 came from the vaccine trials. Gluten challenges evolved over this period of time. And now we're finding that we have a large number of therapies for celiac disease that are in clinical stages of development over the last 5 to 10 years. And one of the ones in terms of the history of that therapy is MTX-101 from Mozart, which Jason will talk to us about. So how do we develop a therapy? Therapies have to pass through multiple phases of clinical trials. Preclinical is where the idea is thought up, where it's tested in the laboratory. Phase 1 usually has 10 to 20 people, phase 2, 100 to 300 or thereabouts, and phase 3 has thousands of patients involved in the trials. We're always interested in the safety of the drug. The benefit has to outweigh the risks. The dose is usually sorted out by phase 2 and becomes less important by phase 3, and I'll briefly give you an example of that today. And then we're interested in how well they're working, and phase 3 is really designed to try and work out exactly how well these drugs are working, and then they become available. Process can take 10 to 15 years and can cost between $1 to $3 billion. So this is an exciting slide, I think, for people with celiac disease to see how advanced these therapies have become over a reasonably short period of time. We now have a lot of drugs in various stages of development for celiac disease. The great majority are in phase 1 or have passed that into phase 2. One got to phase 3, though then didn't progress further, and others are at that early stage of phase 3 now. In clinical trials, we're interested in how well the drugs work, and one of the ways we can assess that is how well the bowel improves. So this is a diseased bowel. This is a healthy bowel with celiac disease. You can see all these beautiful little villi poking up from the lining in the bowel. And this is what it looks under the microscope. So very flat villus to villus easily identified under the microscope. But we also use interleukin-2 now to look at the immune response. So if we give people gluten, how much interleukin-2 are we seeing? How active is this immune response in celiac disease? I briefly wanted to point out the ALV-003 study. So that one from 2014 that I mentioned had 41 people in it. Bowel histology was the primary endpoint. In that study, people took 2 grams of gluten, so under a piece of bread, daily for 6 weeks. But they also took the drug, ALV-003. And they found that those in the placebo group deteriorated in terms of their bowel histology, but not the people who were taking the therapy. So they went on to another study of nearly 500 people. In this study, they decided not to give a gluten challenge. A lot of these people were not in mucosal remission. Their bowel had not healed. So they thought if we give them the drug, or a placebo, those with the drug sure will improve, and those who are on the placebo shouldn't. Unfortunately, everyone improved in that study, so there was no proven benefit of the drug. But it just shows how hard these studies can be to design as they go through those clinical trial phases. Because what probably happened with this trial is when people went into it, they improved their gluten diet. So they all became more gluten-free. So everyone's bowels improved. And when they went back and looked at that study, they found that the symptoms who were taking the higher dose probably did improve in terms of symptom improvement. And this was the last study from 2022. And again, a 2-gram gluten challenge with the drug. And over here on the right, you can see in red where people on placebo deteriorated in their bowel histology, but those taking the drug did not. And this explains how it works. And if you remember those peptides that this drug's trying to break down, if the person in the study is eating a gluten-free meal and we give them breadcrumbs and then give them either the active drug or placebo, if the active drug is working, those peptides will break down and we won't be able to detect them in the urine. And that's what they saw. That in the red, this is the placebo group, so they were able to detect high amounts of those active immunogenic peptides in the urine. But the people who had taken the drug to break down the peptides, they couldn't detect those in the urine. This is a transglutaminase 2 inhibitor, or if you remember that Pac-Man that I talked about, that thing that changes the gluten to become quite immunogenic. They took this daily in different doses because it was a phase 2 study. So they're trying to work out which dose is best for 6 weeks. And they found that in each of the dosing groups, the small bowel was more healed or improved more so than those who were taking placebo. This is a study recently published that we were involved in. It was looking at INU-856, a small molecule that targets SIRT6. And what it does is it heals the bowel wall. So it reduces the permeability of the gut, but also leads to a healing of the cells. And this was a first in human study. So they had 71 healthy people without celiac disease. And they had 6 cohorts. So they gave these healthy people 10 milligrams. And if that was safe, they went to 20 milligrams. And they went all the way up to 160 milligrams. And they did that for each of them once. And then they said, right, well, in part B, we're going to give them different doses. We think these are the effective doses. And we're going to give it to them every day, but for 14 days. And they did that and it was safe. So then in part C, they moved on to people with celiac disease. And they said, let's give them 80 milligrams or 160 milligrams or placebo. So this is what it looked like. So they had their initial bowel assessment at the start. They then were treated with the drug or with placebo. They then had a gluten challenge to see if they could get that bowel to deteriorate. And then they repeated their bowel biopsies to see what it looked like. And this was a phase 1 study, so they're not really designed to look at how well it protects the bowel or how effective it is. But the drug was thought to be very safe and well tolerated by people in the study. And they also found that the bowel seemed to be improving in people who were on the drug. So there was less mucosal deterioration in the small bowel for people who were on gluten who were taking the drug than those who weren't. And finally, just wanted to mention this study as well. So the technology is getting very clever, as you'll hear from Jason shortly. But what they did in this study is they took those toxic gluten peptides and they put them in a little nanoparticle. And then they infused that straight into the blood. And these nanoparticles go to the liver and the spleen, and then the body actually recognizes these and tries to retrain those T cells to say these peptides are actually not dangerous at all. You can tolerate these. So it's trying to restore immune tolerance to people with celiac disease. So they had an infusion on day 1 and a infusion on day 8. Then they had their gluten challenge at week 2, where they had 4 pieces of bread or 12 grams of gluten. And then they had 6 grams of gluten for another 11 days. They looked at their immune response at the start and the end, and they looked at their small bowel biopsies at the start and the end, as well as all of their celiac disease symptoms and safety parameters. what they found was the immune response was much greater in people who weren't on this TAC-101. So when they assessed their immune response post-gluten challenge, people on placebo responded more vigorously than people did who were actually on the drug. And you can see that on the left, the pink going up in people on placebo and not much change in the blue, which is people that are on treatment. And on the right, you can see the red boxes decrease, and that's showing that the small bowel actually deteriorated in people on placebo, but it didn't deteriorate in people actually taking the drug. So you can look yourself. You can go to clinicaltrials.gov where all of these trials are registered before they're performed, and you can look at where they're being performed and how many people they need. So you can participate in a clinical trial. You can help advance celiac disease research, or you could join our celiac research network where you get regular updates about what we're doing. I will just make a quick note as well, because I don't think it's going to show up, that we set up a clinical trials unit in North Queensland as well. And I know people on the talk tonight are from Mackay, which is where I visit. So if you want to be involved in celiac disease studies, you can visit the Coral Sea Clinical Research Institute up there as well. So in summary, why do we need a treatment? We need a treatment because a gluten-free diet alone is insufficient. How do we find one? By continuing to understand the pathophysiology of celiac disease. We now have a large number of potential therapies being trialled, and we'll move forwards as we continue to progress through well-designed and robust clinical trials. So hopefully that's given you an insight on where we're up to. I'd now like to introduce Dr. Jason Chien. So Jason is the Chief Medical Officer at Mozart Therapeutics. He brings nearly 30 years of experience in respiratory infectious diseases from his work in academia, healthcare, and the biopharmaceutical industry. He's a clinician, he's a physician scientist with a passion for improving the lives of people with disease, working across many disciplines, including respiratory infections, pulmonary fibrosis, and autoimmune disease. And Jason's now going to share some of his perspectives on therapies advancing celiac disease. So thank you, Jason.
Well, thanks for that generous introduction, James. And I want to thank the Wesley Research Institute as well for this opportunity to share a little bit about what we're doing at Mozart Therapeutics in this area of celiac disease, but also autoimmune disease. And you'll see that as I share with you some of the details of our science, why we're excited about this approach, which we believe has some applications to all autoimmune disease. So, you know, in the following slide, my first slide, what I'm gonna do is start with a story here. And, you know, James got into it a bit earlier, but I'm gonna go back just a little bit further in terms of talking about the origins of autoimmune disease, but also immune diseases such as celiac disease. So as was mentioned by James, HLA is very important as a risk factor for developing celiac disease. We believe that about 40% of the population actually carry the HLA risk factor for risk for developing celiac, for instance. But why is it that only 1% of that population ultimately develops celiac disease? There must be some other variable. For some of the autoimmune diseases, for instance, type 1 diabetes, multiple sclerosis, it's a viral infection has been implicated as that second factor, that environmental factor for developing the subsequent autoimmune disease or immune-mediated diseases. And what happens is when you get a viral infection, this results in a very robust immune response in your body, and that involves these T cells that James was talking about. And we look at both CD4 as well as CD8 T cells, and we call them pathogenic CD cells. They're important because they are out there scouring our bodies to find the virus to kill, right? Now, for the most part, in this image at the in this image to the left, you'll see that for the most part, the virus infection is controlled, and subsequently there are no issues. However, in an alternative pathway, which is the minority of the time, there are also two other alternative possibilities, all of which stem from the same biology, which is you develop an autoimmune response, an immune response that cross-reacts with the virus as well as a protein in your tissue, an antigen in your tissue, that results in what we call autoimmune disease, or I'm sorry, an autoimmune response, right? Now, if your immune response is working as it should, there's a population of cells that we're very interested in, the CD8 T regulatory cells, or Tregs, shown in blue here. If they are working properly, they will control these pathogenic T cells and prevent that autoimmune response or that immune response to the protein. However, there are individuals out there where these Treg cells, CD8 Treg cells, are not working properly. And in that situation, an autoimmune response can occur. And this ultimately results in an autoimmune disease. Now, a very famous immunologist in Stanford, his name is Mark Davis. We've worked closely with him to work on our program, but he published this data to the right of this slide, where he was able to demonstrate that these CD8 T regulatory cells are what we call upregulated. There are more of them, not only after an infection, but you can also see that they are increased in various different autoimmune diseases, such as celiac disease, type 1 diabetes, multiple sclerosis. So we know that these cells are relevant. But what he also discovered is that even though they're there, they're dysfunctional. And because they're dysfunctional, in animal models, he's demonstrated that this could lead to diseases like celiac disease or autoimmune disease. So that gives you a background of the pathogenesis and the biology of CD8 T regulatory cells and why they're so important. So in my next slide, I give you a schematic of essentially what James has already talked about, right? These CD8 Tregs are fundamental to controlling these pathogenic T cells. And they're important because this is very upstream. This is upstream to all the rest of the damage that occurs, right? So it's these pathogenic T cells can release inflammatory cytokines. They can stimulate B cells to produce autoantibodies, and all of those things go and destroy the tissue that they're targeting at. In the celiac case, this would be the gut epithelial cells. Now, in the next slide, what I show you is how our drug, MTX-101, reactivates these CD8 Tregs to kill the pathogenic T cells. Because the dysfunction is that even though these CD8 Tregs are around, they are not functioning. They're not finding these pathogenic T cells to kill. And because they're not destroying these pathogenic T cells, these T cells are going aberrant, causing more inflammation, activating more B cells, and causing antibodies to react. So if you can go to the next slide. A molecule that's really important to also understand is what we call KIR. And KIR is a killer inhibitory receptor that's on the CD8 Tregs. And what these KIR receptors or these proteins do, KIR proteins do on the surface, is that they block the CD8 Tregs' ability to recognize the pathogenic T cells. So we've designed a drug called MTX-101, which is a bispecific antibody, meaning that there's two heads on this antibody. It binds to the KIR so to free up the KIR from blinding the CD8 Tregs and allowing them to recognize the pathogenic T cells. But we also made it very specific to the CD8 Tregs by adding an extra arm that is a CD8 recognizing arm, so that this antibody is in a very specific way only targeting the CD8 T cells. Now, why is this so important? Because there are other antibodies that do similar things but because they're not specific to CD8 T cells, they could actually activate other cells in your body and cause side effects and cause immunosuppression, which we do not want to do. Now, if you go to the next slide, I'll give you some data in terms of how our T cells work. So first of all, MTX-101, we know restores the CD8 Treg function and can kill these pathogenic CD4 cells. Now, on the left, what I show you is that our drug, as intended, is very selective in terms of how it kills or how it targets these pathogenic T cells. And here I mentioned CD4 T cells, but they can actually also kill CD8 T cells, which are relevant for other autoimmune diseases. I also show on the right that the drug MTX-101 can actually reduce the amount of inflammation when it's present and blocking or inducing the killing of these pathogenic T cells so that they cannot release inflammatory cytokines. And in both these experiments, this has been done using blood specimens that we've collected from celiac patients, where we incubate their blood with and without MTX-101 to demonstrate these effects. Next slide. Now, one very important aspect of this is the selectivity, and I show you that here on the left, where you can see that the green line that goes up demonstrates that our drug binds very tightly to the CD8 T regulatory cells, but it does not bind at all to the other the non-KIR CD8 T cells, the other CD8 T cells that are circulating in your body, and also the other CD4 cells, which are in your T cells that are in your body. So again, the specificity is really important. And why is that important? That's shown in the right panel, where I show you that we've done experiments where we've incubated a patient's blood with our drug, but also with, say, bacterial products, and demonstrated that in the presence of MTX-101, there's no change in terms of your ability, the patient's immune ability to respond to bacteria and also viruses. We've done this with many different types of bacteria and viruses, so that there is very little risk for side effects such as immunosuppression and infections that have been associated with other approaches that are targeting, say, cytokines or other T cells. So in the next slide, my final slide, James talked a lot about all the different approaches to addressing the celiac disease. We believe that all these approaches are important because this is a complex disease, right? And we're tackling this from one angle, but all these different angles are probably going to be needed in some way or fashion, and this is not a zero-sum game. There's a possibility, for instance, in the future that you might require a combination of different drugs to get the maximum benefit. But we do think that targeting CD8 Tregs has a lot of potential advantages. I mean, the first is, it's very selective. So again, unlike approaches that will throw a blanket on the inflammation, for instance, which is often the case with a lot of autoimmune diseases, that causes side effects like immunosuppression and opportunistic infections. Our targeted approach should not result in that. We also think that because we're going after the root of the problem, which is killing of the pathogenic T cells, this approach may result in more durable and hopefully disease course changing effects, to actually allow the target tissue of the immune response to heal. So in our situation, in a celiac situation, this will allow the epithelial barrier in the gut to heal, and also be more or less permeable to these peptides. And then finally, this may not pertain to celiac, but there are now therapies that require cell transplantation, or they're called CAR T-cell therapies. And those therapies, while they have some very promising initial results, are also demonstrating to us that the cells that they transplant into the body might actually change their phenotype, especially in inflammatory situations. And we've been able to demonstrate that because our cells are, we're working with cells that originate in your own body, they are very stable and do not change their phenotype. And that is very critical, because the alternative phenotypes that the allogeneic cells change into are actually turning out to be pathogenic and may actually result in more severe disease as opposed to helping the disease. So I'm going to pause there and entertain any kind of questions that they may be in the audience.
Rightio. Thank you, Jason. I might just, we do have a couple of questions coming through, but I might just start to try and summarize all of that information. You can correct me if I didn't quite get it. But in celiac disease, we have gluten that's broken down in these peptides. That comes across. They get presented to these CD4 T cells. Then they all proliferate. They upregulate, but they're pathogenic. They're not supposed to be there. So people with celiac disease have this really small, long-lived population of these CD4 T cells. And then when they get exposed, they get all these T cells. And then you're saying the CD8 T cells come in, and they're supposed to turn them off and tell them not to worry about it. It's all okay. But because of when the disease started with these viruses, those CD8 T cells got confused and they're not working properly anymore.
Yeah.
Is that what you're saying?
That's the theory. So I mean, you know, the viral theory is very interesting. You know, I mean, this was very important even in COVID. You know, it's a great example actually, because as the COVID virus, SARS-CoV-2, evolved in the human population, you got to remember, you know, a virus's sole job is to survive in the human body so they can proliferate. And the way to do that is they mutate themselves on a random basis, right? And the more they mutate, the higher the likelihood that they will come up with a protein that kind of looks like something similar to something in the human body, which allows them to hide, right? Because then your body can't recognize them. But if this mechanism goes awry, or you're just out of bad luck, one of the proteins that the virus comes up with looks like something in your pancreas for diabetes, for instance, like beta cells or insulin, right? Then you get cross-reactivity. Now, even with that, the CD8 Tregs, if they're working as they should be able to deal with it. They'll say, oh, you know, we've got activity that's against our own tissue, we need to stop it. But it's been shown now that there are, you know, I spoke about KIR, for instance. There are genetic mutations or copy number variations in KIR that are associated with higher risk for autoimmune disease. And why is that? Because if you're expressing more KIR, then you're blinding the CD8 Tregs from these pathogenic cells. So it's very complex biology, but all the pieces are starting to fit together.
And the MTX-101, that's specific for CD8 cells that really are directed against these CD4 cells that are activated with gluten peptides. Is that right?
Yes. Yeah. So it's very targeted. So the CD, so we've taken blood from celiac patients, right? And we have found these pathogenic cells. And they're actually the ones that release IL-2, for instance, that you spoke about. One of the great things about the Immunosanti program is that although the product that they were developing didn't work, they did actually have a side product, which is a diagnostic that might ultimately be approved for or cleared for diagnosing celiac based on this IL-2 response. And our hope is that MTX-101 can shut off the IL-2 response completely by killing the pathogenic CD4 cells in a very specific way.
Yeah, it's fascinating science, Jason. Congratulations
Yeah.
on
Well, you know, we're standing on the backs of giants. I mean, having the opportunity to work with the previous CEO from Immunosanti was a huge privilege because they've learned so much from those clinical trials that they had conducted, right? And they've shared with us what are some of their learnings so that the next company could be successful.
Yeah, and I think for Australians involved in those trials, it's important to realize that it's their blood that's still being looked at now.
Yes. No, it's all the work of the patients that's paying off.
Yeah.
And this is how science works. You build on each other's learnings.
We have a few questions actually that's coming in now. So we might just try and address some of those. So
yeah.
One is in terms of viruses, Jason, that could be a trigger for celiac disease. So we know that reovirus and rotavirus possibly have been indicated.
Yeah.
One of the questions is on Helicobacter as well. Do you have any? My understanding is Helicobacter is protective, but
I'm not aware of, and well, you probably know better than me in terms of Helicobacter. I'm not aware of any associations with or biology with Helicobacter and celiac.
Yeah, no, I don't think it triggers it. What else have we got? Right, if you're attempting to reduce the activity of a specific type of immune cell, what are the implications of this?
Yeah.
In other words, why does the body, I've just lost it.
Yeah. Well, maybe I can anticipate,
that.
you know, why does your body have CD8 Tregs? So this is really interesting, you know, why do CD8 Tregs exist? Well, I think we figured that out in terms of the work that Mark Davis was doing. The interesting thing is that these CD8 Tregs, so we're in phase 1 study right now, and we've already analyzed some of the data from our healthy volunteers. And we've actually done that also preclinically. That's published in our work. You know, in healthy individuals, these CD8 Tregs are there. They're not upregulated, but they're definitely there. And you can by manipulating them or whatever, get them to actually react, right? And that's really interesting. So because what this is telling us is these CD8 Tregs, they're basically sitting dormant, circulating in your body, just kind of monitoring. And what Mark Davis has described is that they're dysfunctional in people who have autoimmune disease. So chicken or the egg thing, right? We believe the hypothesis is that because they're dysfunctional, you develop an immune disease, autoimmune disease, right? So what we're doing is we're hoping with MTX-101 to restore their function. So the beauty of that is if you're restoring a cell to its natural function, the potential side effects is much lower. We're basically allowing them to do what fulfill their life's intention, right? That to help to make sure that they can go back to doing what they're supposed to do, which is survey for pathogenic cells.
Yeah. I mean, we certainly see that with other diseases. So when people are worried about being immunosuppressed with certain drugs, but when we put them on these drugs and their bowel gets healthier, they deal with infections much better because it seems like the immune system's not being distracted.
Yeah.
We've had several questions, Jason, just about I have celiac disease, but I'd like to be in a clinical trial, but when I go into them and I'm given gluten, I get horrifically sick. Some people are passing out, some people vomit.
Yeah.
And I think that's due to the cytokine release, we think. So they get this almost like people with COVID used to get, they'd collapse at the end with this cytokine storm. And you can think of celiac disease a bit in that way, but to a much lesser extent. You get this all this cytokine pro-inflammatory release, and they get these profound symptoms. Without going into specifics, are there ways for studies to be done where you don't have to be exposed to gluten?
Yeah. Yeah. What comes to mind is a paper that was published, I think it was July of last year, out of Finland, where they studied a group of and this is without a drug, it's not a clinical trial, but they took a group of celiac patients and they put them on a gluten-free diet, and then they looked at the immune profile of their blood on a serial basis over a period of, like, I think it was 6 months or 12 months or something like that. And it was really interesting because if you follow that immune profile, what you see is that within about a month, they start to establish a new kind of uh, new immune balance in their bloods, in their circulation, meaning the T cells have come down, and there's a very steep curve in terms of how the T cells come down within that first month, and then that slope gets much more gentle, meaning that by the end of a month, you've essentially arrived at this new homeostasis that you're probably going to be living with if you stay on gluten-free diet for 12 months. So what that tells me is, if you go on a gluten-free diet within a month, you've gotten most of that benefit that you're going to get. And if you stay on that gluten-free diet, yes, you'll slowly get better, but maybe you've kind of plateaued in terms of the amount of benefit you're going to get out of gluten-free diet. And this might explain why a lot of people on gluten-free diets who are very strict still have symptoms. Now, what they proposed, though, was really interesting. They thought that studies that introduce gluten may not be necessary, that you can actually, within a month of treating in a trial, you should be able to see this immune change in the blood to see if a drug is beneficial or not. And that's if you're targeting, the right mechanisms and all that. And for us, this was really interesting because we're interested in these T cells. And it suggests to me that we might be able to design a shorter study without using gluten, without using a gluten challenge, perhaps. But it's all about selecting the right patients. Maybe it's selecting patients who are still symptomatic despite a gluten-free diet. Or maybe it's not as severe of a gluten challenge as has been done in the past, multiple days or that sort of thing. So we have to think about the science a little bit. This is new science, but I have a feeling that clinical trials might start to look a little bit different in the future for celiac disease.
Yeah, very good. We're probably running out of time, but someone's asked you specifically a question here. So, hi, Jason. Would this treatment mean someone with type 1 diabetes and celiac disease would have a lower chance of getting another autoimmune disease?
Wow. I can, we believe this is
Don't want to put you on the spot.
I need to make sure that I don't, it's not that I don't want to say things. It's that I don't want to get people too excited without real data, right? But I can tell you that if you go and read that paper that I talked about from Mark Davis, and it was published in Science 2019, he believes this mechanism is universal in autoimmune disease. There are over 100 autoimmune diseases. He's demonstrated that these cells are upregulated in at least 10 of them. So if you receive this treatment for one autoimmune disease, could it decrease your risk for another? Possibly, but we have to prove that. This is why we have to do these clinical trials that, like James talked about, phase 1, phase 2, phase 3. These are the sort of questions we're asking, right? So, I mean, I'll share with you my thought process. So the first thought process is, first thing we need to do is identify diseases where we believe this drug has the best chance for success. It's no secret, if you go to our website, we're working on celiac disease and type 1 diabetes. And it's because all of our data support these two diseases. We believe there's great evidence for pursuing these diseases. But if and when we demonstrate that there is a lot of potential for benefit in these two diseases, you bet we're thinking about other diseases as well, right? We'll have to do the right trials to that are required by the regulatory agencies to demonstrate that, but and hopefully seek approval for other indications as well.
Great. And pregnancy. So with pregnancy, sometimes we see high-risk people, so first-degree relatives of people with celiac disease.
Yeah.
And they don't have the disease, and they fall pregnant, and then once they've delivered the baby, sometimes they develop celiac disease later on. So it's a change in their immune response, a little bit like getting an infection, because we know the immune system changes to keep that baby inside and then upregulates again. Do you have any thoughts about how this all plays into the development of celiac disease after pregnancy?
Yeah, the first thing I'll just say, I'm not an expert in terms of pathogenesis of celiac disease overall. I mean, James, you're much more of an expert than I am myself. I understand the disease from a T-cell perspective. What I do know though is that the mother's immune system definitely changes after pregnancy, because you're being exposed to an antigen, the baby's antigen, during birth, and that could alter your immune system. I suspect the mechanism is not that different after pregnancy as opposed to after a viral infection or something else like that. It all goes down the same pathway. But that's just my hunch.
Yeah. Maybe we'll just bring it back to something trial related. So there's quite a few questions now coming in about the timeline to go from,
why does it take so long?
Great ideas, great science to actually a drug.
Yeah.
I guess I mentioned 10 to 15 years, and that seems to be what it's playing out. But can you give us some insights from where you're sitting, someone who's developing a drug? Like, what's your thoughts about it?
I love this question, because especially now when artificial intelligence, AI, is taking center stage on a global level from a drug discovery perspective, at least in the United States anyways. It's in the news constantly that there are all these biotechs popping up because they're leveraging AI technology and machine learning to develop new molecules and all that. What I think the industry doesn't talk a lot about is where the bottleneck is. So if it takes 10 years to develop a drug, what do you spend the majority of your time doing? What during that 10 years? Well, I can tell you it's not discovering a molecule. That's usually the first 2 to 3, 4 years. The rest of the time is clinical trials. And why is that? I mean, clinical trials are done basically by brute force, right? There's no machine learning AI that's really going to help us convince a patient to come into a clinical trial. I mean, people are definitely trying, right? They're using social media, they're trying to decentralize clinical trials and all that. But the reality is why does a patient sign up for clinical trials? It's because they hear about the science in forums like this, and they actually believe in it, or they feel so strongly about giving back that they want to participate in trial. And it's also the interactions between physicians like James, who are passionate about the disease, and their patients. It's that interaction that brings people into trials, and that's a very, very labor-intensive process. And then, of course, clinical trials is not real world, right? Out of 1,000 celiac patients, how many will actually participate? Not just volunteer, but how many will actually qualify for a trial? Because trials have different criteria that requires that's required for participation, and some people might get excluded because of those. And those are the things that AI is probably not going to solve for a long time, and that's why trials take so long to enroll. Oh, and then the last thing actually is, sorry, just from a regulatory perspective, they want you to, you want when a drug gets approved to be put on market, you want to make sure that drug is, one, safe, but also has true benefit, efficacy, right? And some of those things take a long time to prove. In celiac disease, for instance, demonstrating that the gut mucosa has healed as a result of being treated by a drug, it takes a long time, 1 to 2 years before you can demonstrate that. So imagine if you're enrolling a trial with 1,000 patients, and every one of them has to be followed for 1 to 2 years. It could take a while.
Yeah. So I guess what you're saying is a lot of intent. There's a lot of interest in developing treatments for celiac disease.
There is.
But at the end of the day, you put the studies forward, but if people aren't joining the studies, they won't progress.
That's right.
That's good. Yeah. So
That's
I think,
right.
The audience today is probably here because they are really interested. And my experience has been that a lot of people who volunteer to be involved in celiac studies are not really doing it for themselves, because a lot of these studies have been very early. So they're really doing it for their relatives or their grandkids,
Yeah.
who may have acquired some of these genes that we pass on that potentially could affect them down the line. That's been my experience with people volunteering. And I think with a lot of people asking, I don't want to get sick. And I think we've refined how much gluten is dangerous or makes people sick in these gluten challenge studies when they're needed, and they're not always needed. Not every study has gluten challenges in them. So there are studies that you can be involved in where you don't have to have a gluten challenge as well, and you can still participate.
Right. Yep.
I think that answers that last question. Celiac disease hereditary. It's very much hereditary. There's quite a few questions about the clinical aspects of celiac disease, which we can answer. So if we're talking about a trial for you guys, you really have to go into phase 1 first. You're doing all your preclinical work, go into phase 1, get that one done as quickly as possible, and then depending on the results, look to phase 2 and beyond.
Yeah. I mentioned before, we're in phase 1 right now. We're just about to get into patients.
Well, I might just see if anyone wants to pop any more questions through specifically for you, Jason. I'll just answer one that's come through. How do I know if my gut is healing? In terms of a clinical question, generally we have to biopsy the gut to know if it's healing. Your blood tests can fall. That doesn't correlate really well with healing in the bowel. And if you go back and look at one of those slides that I presented later, we think one of the problems or the challenges with the gluten-free diet is a lot of the time the gut doesn't heal, despite really strong adherence to a gluten-free diet. We never get people back to what their gut would be as if they didn't have celiac disease, which is why one of the reasons why we're looking at all these other therapies to try and help with that. And yes, I've had a question from a comment from a doctor. So if MTX-101 potentially helps resolve any or all autoimmune diseases, not only celiac disease, that would be handy, as many of my patients have more than one. That's very true. Thyroid disease is very common in celiac disease. Other autoimmune diseases run with celiac disease. They all tend to run in clusters in families.
The term we like to use is we are antigen agnostic, meaning that antigen drives autoimmune disease, and different autoimmune diseases have different antigens that drive them. We don't care what antigen it is. we know that those antigens activate these self-reactive pathogenic T cells, and we kill those T cells.
That's great. We might have to wrap it up now. So just wanted to thank everyone for attending tonight. Very much want to thank Jason for your time and coming along and speaking to us.
Thank you all.
And everyone at Wesley Research who's put in a lot of effort to get this happening. What we might do is we might later try and collate some of these questions, Jason. We might put them to you to answer in a blog or something that we can post later on for people
Sure.
that haven't had their questions answered. But yes, thank you very much. I'll just try and share the last slides by reducing this. Hopefully people can see that. But we might wrap it up, and thank you very much for attending. And hopefully we'll see you at our next webinar. If you'd like to also email us or get in touch about topics you'd like covered clinically, it might be about oats or it might be about gluten challenges, let us know, and we can certainly try and work out what's of most interest and put that forward for the next presentation. So thank you very much and good evening.